Hi my students, my name is Rukia Abdallah. I'm a biology teacher. I'm going to discuss with you about transport. I will concentrate myself in the subtopic of transport in plants. Okay, last time we saw transport in plants based on vascular tissues. We described the structure of vascular tissues, that is xylem and phloem. We also discussed the adaptations of each vascular tissue to its function. Therefore, today we are going to discuss another aspect. We are going to cover the so-called paths of water and mineral salts across the roots. And after that, we will also discuss the forces governing upward movement of water. But before doing anything, it is better to start with the question number 1. Question number 1 based on the active and the passive transport. The question says that distinguish between active and the passive transport. You are required to distinguish between active and the passive transport. Mind you that you cannot make any kind of distinction without knowing what is active transport and what is passive transport. Therefore, you are required to define the terms first. After defining the terms, then you will be in a better position to differentiate the two. Here, I'm going to start with the passive. Passive transport. Mind you that we saw the vascular tissues and we said that for the case of higher organisms, they should have the transport system which will enable materials to be transported from one point to another. For that case, materials cannot move without having these two, that is passive transport or active transport. When you deal with the passive transport, it means we consider movement of materials following concentration gradient. It means they move from the area of high concentration to the area of low concentration. That is passive transport. But apart from that, energy is not employed. Energy is not employed in a passive transport, and the process is slow. When I say slow here, I mean that movement of materials is not rapid compared to active transport. And when we compare its efficiency with the active, this one is less efficient. Less efficient. And point number 4 is it is by direction. Movement is by direction. I think you understand when I say movement is by direction. It means it can go to one side, but when the concentration there becomes big, then it can change to the opposite direction. That is why we say that movement of material is by direction. And there are about 3 types of passive transport. The first one is known as osmosis. Osmosis. I think you know what osmosis is, because you have covered that one in O level and even in A level in Form 5. You discussed about osmosis. For your information, osmosis involves movement of water. Movement of water from the area of high water potential to the area of low water potential. I repeat, I said that osmosis here, based on movement of water only. Water moves from high water potential to the area of low water potential through semi-permeable membrane. The membrane should be there in order to allow materials to move from the area of high water potential to the area of low water potential. Another one is diffusion. Diffusion. Diffusion involves movement of materials from the area of high concentration to the area of low concentration. But don't you mix with the osmosis. I said that osmosis involves movement of water. But other materials apart from water move by diffusion from the area of its high concentration to the area of low concentration. In both cases, energy is not needed. That is why it have been categorized to the group of passive transport. Number 3 is facilitated diffusion. Facilitated diffusion is some case of diffusion, but here it involves carrier molecules. For example, you may have 2 medium. Let's say this is medium A and here medium B. These 2 medium have been separated by the membrane. And here, let's say there is high concentration of ions compared to this side. But without carrier molecules, these molecules cannot diffuse to this side. So, the membrane here have carrier molecule. Let's say this is a carrier molecule. When it appears in that shape, it attracts these molecules here. And when the molecules are attracted to this side, the shape of the carrier molecule changes. Instead of opening to that side, it now opens to this side. And when it opens to this side, it allows the molecule to move from side B to side A. This is also passive diffusion. I mean passive means of transporting materials. Energy is not required. And apart from passive, there is also active transport. Active transport allows materials to move against concentration gradient. When I say against concentration gradient, it means materials move from the area of low concentration to the area of high concentration. You may take an example of digestive tract. You always eat food. But regardless the concentration of food in the digestive tract, still materials can move to one direction only, whether the concentration there is high or low. If the concentration in the digestive tract is low, obviously active transport will be employed in order to push material from the area of low concentration to the area of high concentration. Okay, after knowing the meaning of passive transport and the active transport, now you will be in a better position to make distinction or to differentiate the two terms. You may differentiate the two terms. For example, you may say that passive transport is less efficient compared to active transport. That is number one. Second, you may differentiate by saying that passive transport does not require the use of energy, but active transport use energy. Number three, you may also consider whether the materials moves to both sides or only one side. When you use that criterion, you may say that passive transportation is bidirectional, but active transport is unidirectional. It goes only in one direction, like a burette. When you fire it, it goes to only one direction. Okay. Another else, active transport is more efficient compared to passive transport. I think you have understood. You may also check your notes of Form 5. You also discussed the passive transport and the active transport in cytology. Therefore, it is better for you to revise each and everything concerning with the active and the passive transport. Question number 2 says that describe the paths of movement of water and mineral salts across the roots. Paths of movement of water and mineral salts across the roots. Let's say this is the structure of the root. This one is root hair. These are soil particles. The root here is composed of different kinds of cells. For instance, there is the so-called epidermal cell. There is also cortex. Cortex cells. Apart from cortex, there is also endodermal cells and pericycle cells. The cells have been arranged following that direction, from here to this direction. We expect to see epidermal cells here. After epidermal cells, cortex cells follows. After cortex, endodermal cells, then pericycle. After reaching to the pericycle, we expect then materials to move to the xylem vessel. So the question says, describe the paths of movement of water and the mineral salts. Last time we saw that water comes from the soil. It comes from the soil here, water. That water should be absorbed. Should be absorbed by root hairs. But in order for this cell to absorb water, it means the concentration here should differ. Due to that, water will move by osmosis. Osmosis will take place. Water will be drawn from soil to epidermal cells. When water enters the epidermal cell here, the protoplasm here becomes diluted due to the entry of water. Due to that, the water potential in this area will be high compared to water potential in the cortex cells. This will make water to move from the area of high water potential, that means from epidermal cells, to the area of low water potential, that means in cortex. The entry of water in cortex also makes its protoplasm to be diluted and acquiring high water potential. When I say high water potential, it means amount of water here will be high compared to solute concentration. Therefore, solute concentration here will be low, but water potential will be high. This will make water to move again from here, cortical cells with the high water potential, to endodermal cells which experience low water potential. And when it enters to endodermal cells, again the protoplasm here becomes what? Diluted, and the high water potential is created. The solute potential becomes what? Low. Due to that, materials will move from here, from endodermal cells to pericycle. When it enters the pericycle, the same story takes place. It means the protoplasm there becomes diluted in case of concentration, and it acquires high water potential. That high water potential which is created there will make water to move from pericycle and enter the xylem. From there, another mechanism will take place. So this is how water and mineral salts move. But mind you that for the case of water, osmosis will take place here. But how about minerals? How about mineral salts? For the case of mineral salts, it depends on the concentration of salts in the first cell here. Sometimes concentration may be high inside rather than the soil water. If concentration here is high compared to the soil water, then mineral salts there will be taken by active means. That is why at the beginning there I introduced to you active and passive transport. And if concentration of dissolved materials will be high in the soil compared to this cell, then simple diffusion will take place. Salts will diffuse to this cell, and the material will continue to move. I think you have understood. Okay, check that diagram. This diagram represents the cells of the roots. Represents the cells of the roots starting from here. Here, the diagram is represented by 4 cells. It doesn't mean that the root contains only 4 cells. There are so many epidermal cells, so many cortical cells, so many endodermal cells, so many pericycle cells. But in order for you to understand easily, it is better to use only these 4 cells. Starting from here, this is the epidermal cell. The epidermal cell is found in the root here, for instance. We expect this cell to draw water from the soil by osmosis, as I have already explained there. And when it reaches here, the contents here becomes diluted. That dilution makes solute potential in this cell to be lower than solute potential in a cortex cell. At the same time, water potential here will be high compared to this cell. Therefore, water will move from the area of high water potential, it means from here, to low water potential. After reaching here, the same story takes place. The cortex protoplasm becomes diluted in terms of concentration, and it acquires high water potential. That high water potential will be used to push water from this cell to endodermal cells. After reaching here, the protoplasm will be diluted. From there, water will be pushed due to its high water potential to pericycle. From pericycle, when water potential is created in this side, then water will be moved to the xylem vessel. But mind you that there are 3 routes through which materials can move in that route. The 3 routes are apoplasty, symplasty, and the vacuolar pathway. Okay, I think you check these arrows, the blackish one, like this one, here. The arrow which passes through this layer, this is the cell wall, So, one route, water and minerals will move through cell walls. From cell wall of epidermis cell to the cell wall of cortex cell, then it enters to the cell wall of endodermal cells. But after reaching here, water and minerals cannot continue to move through the cell walls. Why? Think it. Why? I think you check these bands. These bands are known as Kasparian strips. Kasparian strips are composed of suberin materials. Suberin materials prevent water and minerals to pass through. It means they are waterproofing materials. That is why they force water now to enter the protoplasm of endodermis. This phenomenon is found only in roots. In leaves, no Kasparian strips. Therefore, this route through which water moves through the cell walls is known as apoplasty pathway. It is known as apoplasty pathway. But apart from that, water also can move from one cytoplasm to another cytoplasm. For example, here, these arrows. Water moves from here and it enters to the cytoplasm of another cell, that is the cortex. From here, it enters the cytoplasm of endodermis. From endodermis, it enters the cytoplasm of pericycle. This way is what is called the symplasty pathway. Symplasty pathway. The third way is not indicated in this diagram. The third way is vacuolar pathway. Let me show you another diagram in order to visualize the vacuolar pathway. In this diagram, all the three routes have been indicated. That is apoplasty pathway, symplasty pathway, and the vacuolar pathway. You see this arrow here at the middle. This arrow indicates the vacuolar pathway. It passes from vacuole to vacuole, from vacuole A of cell A to vacuole B of cell B to vacuole C of cell C. Therefore, when water and the mineral salts move across the vacuola, we call that route as vacuolar pathway. And here, this one moves through the cytoplasm. It moves from one cytoplasm to the second cytoplasm of another cell through plasmodesmata, from cell B to cell C through plasmodesmata. Therefore, this way is what is called the symplasty pathway. And the last one moves here through the cell wall A, then to cell wall B, then to cell wall C. This pathway is what is known as apoplasty pathway. But in this diagram, the Kasparian strips have not been indicated. The reason behind is because the names here which have been given is only A, B, and C. But always remember that when water passes through the cell wall, when it reaches the endodermal cell wall, there are Kasparian strips which have been made up of suberin. It acts as waterproof and it forces water now, which moves through the cell walls to enter the protoplasm of the cell. I think you have understood. And another thing here is that when water enters the pericycle, in pericycle here, when it enters the pericycle, it means it creates secretion of mineral salts into xylem. Therefore, when xylem secretes mineral salts, its concentration becomes high and its water potential becomes low. In so doing, it is easy for water to enter the xylem vessel. So, those are the ways through which water can be transported across the root. Question 3 says that with the help of diagram, describe the paths of movement of water and the mineral salts across the roots. There is no difference between question number 2 and question number 3. Question 3, there is only with the help of diagrams. It means you are required to describe the same paths, but diagram should be used. As your homework, you will do that question. Question 4 says why Kasparian strips are important in roots. We have seen Kasparian strips in endodermal cells in roots. Here we are required to explain the importance of Kasparian strips. Okay, number 1, it regulates amount of water entering the xylem vessel. It regulates, you can say it regulates, so it controls the amount of water entering the xylem. That is 1. Second, it prevents entry of toxins and the other pathogens. Therefore, you don't expect toxins to enter the xylem vessel. That is why materials which move along the cell wall should be pushed to enter the protoplasm in order to make sure that no toxins which will enter the xylem. Apart from that, when water and mineral salts are forced to enter the protoplasm of the endodermal cells, it makes the secretion, active secretion of salts into xylem vessels. It enables active secretion of salts into xylem vessels. Therefore, the contents of xylem vessel will acquire low water potential due to the presence of mineral salts. And that phenomenon is very important in order to enable it to draw water from the pericycle and other cells. I think you have understood there. Apart from that, you may also find that these bands attaches this membrane to the wall here. So, if plasmolysis takes place, still this membrane will be attached to the cell wall because of the presence of these bands. So, these are some of the importance of Kasparian strips in roots. From there, we can move from roots, we go to the xylem. We are going to check upward movement of water in the xylem. But in order to describe that one, we are going to use guidance which have been prepared by your presenter. That is question number 5. Question 5 says that how each of the following forces helps in upward movement of water and the mineral salts. How each of the following forces helps in upward movement of water and the mineral salts. Here, I'm going to revise with you on 3 forces. One is root pressure. Root pressure. At the beginning, I said that water enters the root hair by osmosis. At the same time, I said that when water and mineral salts reach the endodermal cells, there is active secretion of mineral salts into xylem. Due to that, here we expect the protoplasm in the xylem there to create forces. The force will be created there. That force will trap or will pull water from the neighboring cells. At the same time here, osmosis still takes place. So these forces create hydrostatic pressure. Hydrostatic pressure will be created. That hydrostatic pressure is the one which builds the so-called root pressure. But mind you that root pressure cannot account movement of water up to the higher stems. Let's say 300 meters cannot be counted by root pressure. It means another force is needed in order to push water higher in the stem. It is my hope that you have understood. As usual, don't forget to interact with your materials which you have been provided by your teacher. Interact also with your books in order to make notes about the root pressure. Apart from root pressure, there is also capillarity. Capillarity. Capillarity is caused due to narrow tubes, force which is created in narrow tubes. We know that xylem, as we described in the last presentation, the tubes are narrow. The narrowness of the tubes makes water molecules themselves to attract from each other. The force which is known as cohesion, force between like molecules. Apart from that, water molecules also collide with the wall of the vessel. Therefore, another force here is created. That force which is created between the wall of the vessel and the water is known as adhesion force. These two forces makes water to raise up. Water raise up in a continuous column. Because of what? These two forces. This force is what is known as capillarity. But still, capillarity cannot account movement of water into tall trees. Let's say 200 meters cannot be accounted by this force. It means another force is needed. That force which is needed here is what is called the transpiration pull. Transpiration pull originates from above. It means originate from the leaves, and it is due to transpiration. I think you know what is transpiration. Loss of water in form of vapor. That process is called transpiration. Therefore, when transpiration takes place, let's say from mesophyll cells which are found here at the leaves, when it loads here, it makes this area to have low water potential. Water potential here will be low compared to the neighboring cells. Therefore, water will be drawn from here to this cell. And when this water molecule moves from, let's say, this point to here, it makes also another water which is here to be pulled from this area to here. Because of what? We said that because of cohesion force. These water molecules have been pulled to each other. So when one molecule is pulled, the molecule also pulls another water molecule and making it to move in a continuous column. And this phenomenon will go down the xylem and making up water to move up to tall trees. This pulling force is what is called the transpiration pull. I think you have understood. As usual, if you want to master these things, listening alone cannot do anything. You are required to use more efforts. More efforts how? By reading. Sit down, read your notes, read your books. Try to answer several questions. If you stuck somewhere, make sure that you find the solution for it. When you do that, obviously you will master each and everything. Therefore, I beg you, please, 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 make thoroughly revision. As your homework, I gave you question number 3. Don't forget to do that question. Make sure that you do your work. The question says that with the help of diagram, describe the paths of movement of water and mineral salts across the roots. Go and read. I think you have achieved the objectives here. My objective number 1 here, I required you to be able to distinguish between active and passive transport. Therefore, assess yourself. Are you able to distinguish active and passive transport? Objective number 2 is you are required to be able to describe the paths of movement of water across the roots. The paths which I'm talking about are apoplast, symplast, and vacuolar. Now I'm asking you a question. Are you able to describe those three paths? If yes, it is good. And if no, please don't close up your books. Continue to read until you achieve that objectives. Objective number 3 is you are required to explain upward movement of water. Upward movement of water. Now, are you able to explain upward movement of water? That means in the xylem vessel, are you able? And the last aspect or the last objective is to explain forces governing upward movement of water. The forces which I'm talking about are root pressure, capillarity, and the transpiration pull. Now I'm asking you, are you able to explain those forces governing upward movement of water? If you managed to achieve all those 4 objectives, then I can say that my presentation is successful. Thank you for your listening. We shall meet in another presentation. Thank you.